We have done the secondary growth in Dicotstem in great details. Now we are going to see the secondary growth in Dicot roots, if it is seen. Yes, it is seen, and it is quite similar to that in the Dicot stems. Now there are a few points of differences that you have to remember, and we have to see what these are. As you can see over here, the steps are quite similar. One thing is there that no annual rings are formed, and secondly, we have to remember one thing, that the formation of vascular cambium is taking place as the first step, unlike the formation of cambial ring, which was the first step in Dicot stem. Then the vascular cambium shows the activity, which is the formation of secondary tissue from itself, and lastly there is formation and activity of the cork cambium. Now because you are quite aware of what happens in secondary growth, there is formation of secondary xylem and secondary phloem, and we have a presentation as well for this particular topic. What we are going to see in this lesson is that how it is different, and a little bit of my own diagrams to help you out to understand the topic. First of all, you know that in the case of root, we have little bit different organization at anatomical level. If you can recall, we had done the diagram where we discussed that the vascular bundles are radial over here. First thing that is to be noted. Now because the growth has to take place at cambium level, and the cambium is vascular cambium, we are going to be more concerned about the stele in the beginning, which comprises the xylem and phloem and endodermis. Now this is what a stele of root looks like, dicot root looks like. Here we have the layer of endodermis, and endodermis has nothing to do at present. What is important to understand at this step is that the vascular bundles happen to be radial, and there is no strip of cambium. The vascular bundles are not open, no strip of cambium is found. So how would the secondary growth take place? The first point of remembrance is that the vascular cambium, which is formed in the secondary growth in the case of a dicot root, is secondary in origin. That means some originally present cells would become meristematic. They would get de-differentiated and they would become meristematic. And which cells would become meristematic? That is conjunctive tissue. Now try to recall what was this conjunctive tissue, or where were these cells located. So little bit exercise for your mind. If you can recall. If you cannot recall, then I am supposedly providing you with the answer that the conjunctive tissue lied between the oval shaped phloem and xylem. Now such was the case. These were the xylems. Such was the arrangement, right? If somehow you can remember. If you cannot remember, that's bad. You need to learn these things. That's why we are dedicating our lessons to these topics. In case you don't remember that, it's not that bad. You can see the video all over again. This is xylem, and we have somewhat egg-shaped phloem. Few cells of phloem would be present over here like this. Right? Never mind if you can't remember. Even I took many years to remember these things. That's why I'm able to tell you. But I don't have to get marks, so it's better you remember them at the earliest. Diagrams are very, very necessary. That's why we put so much stress on understanding diagrams. Now this is what the stele of a dicot root looks like. Now in between over here there lies conjunctive tissue, if you remember. Now what will happen when we have to see the secondary growth in a dicot root? This region would become meristematic. Alright, this region. This conjunctive tissue is going to become meristematic. Apart from this region, now this is conjunctive tissue as well as the pericycle. Now this part would also become meristematic, and we are going to have some stellar shaped region like this. You're going to find meristematic part. Something like this would become. A ring would be formed. Now this is not a ring, it's a wavy pattern. such cambium would be formed. Alright, something like this. Now what will happen is that this is going to give rise to cells. After some time, this will form like this, a stellar pattern. Now as you know, the cambium is going to give rise to cells inwards and outwards, and with time what happens, the cells which are formed inwards are produced at much rapid rate as compared to outwards. So gradually this particular thing is going to overtake this particular part. This is going to push it more and more inside. The pericycle is becoming parenchymatous, or not parenchymatous, meristematic. So this primary component is going to be pushed inside, and what we are going to have is that the cambium takes up such a shape like this. Now outside we are having phloem and inside we are having xylem. This would be the actual condition of cambium that is formed. Now you can relate with it. Now this is going to produce xylem inside and phloem outside, because xylem is produced at a greater rate. So after some time it is going to crush the primary phloem, it is going to crush the secondary phloem as well, and it is going to form a circular shape which gives rise xylem inside and phloem outside. Now I hope I am clear. If not, I am going to repeat it. See, in the beginning this is phloem, here we had xylem. In the beginning the conjunctive tissue became meristematic because it was secondary in origin. So we need to have some previously present tissue that would get converted to meristematic tissue. Later on it forms this sort of shape, stellar shape. It's not stellar though, but we can relate that. It forms this stellar shape, and it produces in a lower rate the phloem towards outside, and towards inside rapidly it produces the secondary xylem. With time, this particular primary phloem would get crushed, and the cambium that was present like this in the stellar shape, it is going to gradually take circular shape because the phloem is getting crushed slowly and steadily. And why it is being crushed? Because xylem is produced at a much rapid rate. So we have a circular cambium ring which is being produced. Lots of xylem is being produced inside. Which xylem? Secondary xylem, because the pith was not highly conspicuous, so it gets more and more reduced. The phloem gets very much crushed. And talking about the rest of the layers. We have seen the activity of vascular cambium is almost similar to that in the case of Dicotstem. Now we have to talk about the formation and activity of cork cambium. This was the steel part that we had discussed. What about the regions which are present over here? Now you remember that the region outwards to the steel had endodermis, and there were many cortical layers. And after that we had epiblemma. This was all about the Dicot root, right? Now the cells of cortex, few cortical cells, and at the same time few cells of pericycle as well. Pericycle, which is present near the endodermis. In fact, it is the pericycle cells which form the cork cambium. You have to remember, the pericycle cells, they become meristematic. They start producing the cells in the same pattern as it was in the case of stem, and the endodermis, the cortical layers, and the epiblemma. Epiblemma is lost itself after some time. When the cork cambium starts producing the cells, this part is gone. It vanishes. Then most of the cortical layers, they are crushed and they vanish. What we have is, we have the pericycle that acts as cork cambium. It gives rise to secondary cortex towards inside, that would be in close proximity to secondary phloem, and outside it gives rise to cork. So what do we have in the end of a root that has undergone secondary growth? We had the outermost cork. Then we'll have cork cambium that would be formed out of pericycle. There would be secondary cortex with parenchymatous cells. Epiblemma is lost. Most of the cortical layers are lost. Then we'll have secondary phloem. Primary phloem is almost lost, so we don't have primary phloem. We have vascular cambium, which was formed by dedifferentiation of conjunctive tissue. We have secondary phloem, and here also the secondary xylem. Here also the secondary xylem has completely crushed the primary xylem. So this is all what we have in the case of a dicot root which has undergone secondary growth. In case you get to see a dicot root which has all these layers, you would start from secondary xylem in the center of the root, that is the central point will have secondary xylem. As you move outwards till the epidermis, which is lost, completely lost, you're going to find cork layers, which is followed by cork cambium. Then you have secondary cortex, secondary phloem, which is again crushed, vascular cambium, which was stellar in shape in the beginning. It was absent in the beginning. It was completely absent. Later on, it took a stellar shape. Then it took a circular shape. Then it forms the secondary xylem in great quantities. So this is all that you have to understand in secondary growth for a dicot.